Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Effect of Bacteriophages in Reduction of Salmonella on White Meat During Processing. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Phageguard, who developed the content for this presentation. Phageguard contributes to safer food production by using phages. As the natural enemy of bacteria, phages specifically kill pathogens like Salmonella and Listeria and leave the good ones intact. They are green, smart, and easy to apply on food via spraying, misting, or dripping. Phages can also be used directly on food contact surfaces or in the processing environment. Now, I would like to introduce our speakers for today's event. Our first speaker is Linda, who has a BSc in chemistry and an MTech in environmental health, food safety, Central University of Technology. She has been involved in management system consulting, training, and auditing since the early 1990s, when ISO 9001 was first implemented in the food industry in South Africa. She has held lead auditor status with SAA-TCA in food safety and auditor status for quality management systems, and has conducted numerous training courses on HACCP, FSSC 22000, and internal auditing. Linda is the co-founder of Food Focus, a company which aims to educate on the subject of food compliance, supporting everyone across the length of the food chain. Her passion and current field of study is food safety culture. And so now, without further ado, I'd like to hand the mic over to our speaker, Linda. You may begin when you're ready.
Thank you, Ryan, and good morning, everyone, and thank you for joining us today. It's my pleasure to be the moderator for this webinar today. Thank you so much to Phageguard for this wonderful opportunity. And although I'm the first speaker, I'm not really going to be doing much talking. All I'm going to be doing is introducing you to a panel of experts who will be taking us through a number of different aspects relating to bacteriophages in the reduction of salmonella during processing. The structure of today's webinar is as follows. We're going to be chatting to Ms. Najib Salim from RCL Foods, and she's going to be chatting to us about the risk mitigation in the food industry, specifically within chicken processing, from a microbiological perspective. In other words, what are the risks regarding salmonella and other bacteria within the food industry, particularly on the poultry side? We're then going to be chatting to Professor Frank de Vlieckere, and he's going to be talking to us about the use of phages. What is a phage, and how do bacteriophages present possible solutions to some of the risks that are faced within chicken processing? We then are chatting to Professor Peter Goos from the Center for Food Safety at the University of Stellenbosch, where he's going to be presenting a possible solution based on some research that has been done regarding the efficacy of bacteriophages in the reduction of salmonella on chicken carcasses in a processing setting. Finally, we'll be bringing it all to a close when we chat to the CEO of the Southern African Poultry Association, Mr. Isak Breitenbach, and he'll be telling us what the research and the results of this research actually mean to the poultry industry as a potential solution and improvement for food safety going forward. Now, I'm sure you all agree with me that the production of safe food is absolutely paramount. And although there are more than 2,500 different Salmonella serotypes, and only a few that really cause problems in the poultry or white meat sector, when they do cause problems, there are significant problems. We have problems that, or serotypes that affect or contaminate the flocks, which can cause economic damage to the breeders. But not only that, we have serotypes that can foodborne illness within the human population. And so it's essential that we have a good mitigation strategy to ensure that we keep salmonella levels as low as possible. And this involves a number of different aspects. And today we're going to be exploring these and specifically looking at the opportunities for bacteriophages as a possible reduction strategy within this particular sector. So without further ado, it's time to get you involved. And so before I hand over to our first speaker, Ryan, won't you run the first poll for us so we can see who we have out there and who's participating in today's webinar. Thanks, Ryan. Over to you.
Yes, thank you very much. So as Linda says, we've got a poll question for you all to interact with right now. It should be appearing on everyone's screen, and you can choose from the answers in front of you and then click submit. The question we have for you is, what is the primary area in which you work? Your answer options are food processing, academia, government, or human health. Again, the question is, what is the primary area in which you work? We appreciate you taking the time right now to consider an answer and participate in this. It looks like most of you have voted though, so let's go ahead and take a look at those results. Coming in at 69% for food processing, 23% for academia, 6% for government, and then 3% for human health. Once again, thanks for your participation. Linda, it's back to you.
Great. So this means we're talking to the right audience, Ryan. We've got the people that potentially have a problem and could be looking to phages as a solution. To help us to understand some of the challenges that are faced within processing, I'm going to hand over to our first speaker, that being Ms. Najib. Najib is currently employed as the Executive of Safety, Health and Environment, Risk, Quality and Food Safety at RCL Foods. That's quite a mouthful, Najib. You must need a very big business card to hold all of this information. She's worked in the food industry for over 15 years, and she has a business degree as well as science degrees. Her expertise is not just limited to food and beverage, but she has a range of areas of that she's worked in, including red meat, poultry, fresh produce, cereals and grains. She's really involved as a technical specialist. She's worked in training, consulting and auditing services, but currently she's adding value when it comes to strategy, the design and implementation of food safety management system strategies within the RCL Foods Group. Najib, it's great to have you with us, and we're looking forward to your presentation on microbiological risk mitigation within the food industry. It's over to you. Thank you, Ryan.
Good morning, everybody. Before I continue, I just would need to read a disclaimer, short disclaimer. The content related to the Phageguard trials are not that of RCL Foods processing sites. Our participation is to share general information on microbiological risk mitigation with reference to historical publicly available information. Thank you. Linda, you may move on to the next slide. All right. In terms of microbiological risk, there are some key aspects that need to be understood in terms of understanding the nature of microbiological risk that may affect any facility or any processing facility. In this regard, there are three main aspects that need to be assessed: the type of hazard, the source of the hazard, as well as the risk. Now you see that I have actually listed some very, very popular pathogens that we are all aware of, and I'm pretty sure that Phageguard will be giving some insight as to how they have managed to find a method or a solution to try and control some of these hazards. But regardless of the process, the stage in the supply chain, these hazards may occur for a reason. Hence, understanding the profile and the history of the hazard is quite important. I mention history because you will see later on in my example how the nature of the hazard and how it's mutated over time can really result in a crisis. In terms of the source, one would need to understand the nature of the hazard in relation to the source. For example, it can be expected that raw chicken may have, for example, listeria or even certain strains of salmonella, given the nature of its growth. It must be expanded, therefore, to serotypes and those specific serotypes or strains that may have a specific impact in terms of your customer, consumer, that you are trying to reach out to, and in respect to the nature in which you will be selling this product. So the justification for the source must be highlighted. Why are you saying that that is specific source related to this specific hazard? In addition, you have to consider certain elements. The process that you will be following, that is any bactericidal steps that you might have, the product that you are producing, as well as the customer or consumer use. So, for example, the use of a raw product versus a cooked product. If something's to be further processed, there might be a reason why certain microorganisms may still be present. However, handling conditions for that specific raw material must be clearly articulated to prevent any further contamination. Next slide, please. One very important aspect in terms of understanding what your risks are, what your microbiological risks are, is trying to find them. I cannot stress the importance of carrying out the right test methodologies in trying to find microbiological risks. This is often the reason for organizations not understanding that a risk may exist or downplaying the type of risk that may exist. And hence, I'd like to draw your attention to three very important aspects to be considered in terms of making sure that the risk is identified and subsequently mitigated. The criteria for selection in terms of the method used is very important. So any microbiological laboratory that hopefully is accredited would explain to you that there are very different selection criteria in terms of the methods that are used. For example, if you look at Listeria monocytogenes or Listeria in general, there are methods related to ISO, APNO, AOAC methods, and it's important that you describe what is your desired outcome in terms of the microorganism you are looking for so that the right method is actually applied. Also to consider is whether you are looking for the specific microbiological risk in your environment versus your products. And what is the aim of trying to find this microbiological risk? Is it to verify, for example, the efficacy of hygiene, or is it to understand what is the nature of the hazards that may be naturally present in the environment, or is it to verify efficacy of processes or processing aids? The method performance also looks at other aspects such as sensitivity, specificity, and selectivity. It's very important when you are selecting a method as well that you consider certain important factors like turnaround time. For example, if you are requiring a method for product release and you have a very short shelf life, something you need to consider is the turnaround time for that method. A 5-day ISO method versus a 48-hour method, whether it's a virus or whatever the case may be. That is very important because decisions would need to be made in respect to those results that would be coming out. And of course, every business has to consider the cost of the methods chosen. So also very important in terms of the methodologies is validation methods. It's very important that we recognize that the laboratories need to use those methods that are validated for that specific product. So a method may be validated for testing of water, for example, but not necessarily for testing of a baked product or of a dairy product. It is very important that when engaging with the laboratories or even within your own laboratories, you consider the validation method and for what application it was validated for. It must be fit for purpose, or once again, you may be getting results that are not a true reflection of actual microbiological risk. I will not go into details with regard to the sensitivity and specificity, but it's basically based on qualitative and quantitative method testing. It is very important further to identify whether you are interested in just the mere presence of an organism or the volume of that organism that may be there, the quantification. For this, you would be guided by regulations or your in-house specifications that would state whether you are concerned about the absence or the presence of an organism or the level. Where specification exists for level, one cannot be using an absent or present method because it would not tell you whether a risk exists. Next slide, please, Linda. So once you've identified your hazards, it's very important to understand that in terms of microbiological risk mitigation, it all fits somewhere within our pyramid of food safety management systems. As a food manufacturer, you may all know that it is very important that in terms of producing a product that conforms to specification, it starts with, number one, having the right organization cultures and behaviors at place. Some of you may have heard the term food safety culture being used quite frequently in recent times, and that talks to behaviors that support the control of risk within an organization. Hence, it forms the foundation of any structure you may have within an organization. Accompanied by that must be good leadership and management commitment to the process. And as we would all know, any manufacturing site is operating on regulations and legislation that they need to comply with. There are the basic building blocks in terms of what should I comply with. There are basic requirements. So, in South Africa, in example, you cannot operate a food premises without having an R638 certificate, which talks to basic hygiene requirements for that facility and handling of those products. This is the element where regulatory bodies fit in. At this specific stage of legal compliance and regulatory compliance, microbiological risks are often defined in terms of the acceptable levels for different food products. Compulsory specifications for certain products would also define at this stage what is acceptable and what is not acceptable. And in some instances would go as far as defining the test methodologies that need to be used for that microbiological risk. Hence, in terms of mitigation strategies, it is very important to make reference to regulation, to legislation, to legal compliance elements. And where regulations and legislation do not exist, the next step is to look at what international standards or regulations or guidelines documents may exist. For example, where there's a gap in the South African legislation, we often fall to what is written out in codex. And it must be relevant to the product that you are processing. This basic foundation then gives rise to the next stage in terms of your food safety management system, risk mitigation process, which is your prerequisite program, where you would further define in terms of specification. When we look at microbiological specification, you may further define what are the desirable levels that you would like to have in place. Note that these specifications may not be weaker than that which is written down in terms of legislation. They would have to be stricter than existing legislation. And most of us would know that our customers often have either what is equal to legislation or regulations or a tighter specification in that regard. These guidelines, these in-house specifications that you have gotten written down as part of your food safety management systems and your prerequisite programs must be adhered to, and there must be criteria in terms of what would be a go or a no-go area, and what would be the actions to prevent reoccurrence should it occur, what would be the corrective actions in those incidences. So it's not just about identifying the hazard, the source, where in your process it may appear. It's also about testing and getting the results that are aligned to that specific process or product, using the right test methods, and also most importantly, making sure that the outcome is in line with not just your in-house specifications, but also with legislation or regulations that are defined. Next slide, please, Linda. Now, what happens if we fail to comply to these regulations, or if we fail to conduct risk assessments that help us to understand where microbiological risks may apply? Now, some of these may not be microbiologically related risks. However, they are risks that have resulted in recalls across the world, from vehicles to pregnancy tests, to what some of you may have heard of in the recent years in 2018, the recall for processed meats in South Africa in terms of the listeriosis outbreak. Now, the reason why these things may occur can be various. It can be based on processing or manufacturer negligence. It can be based on supplier negligence. It can be based on a break in processes. It can be based on the fact that standards, regulations, or guidelines might not easily articulate what is required in terms of the specification. And for this purpose, I have illustrated that apart from having all of these systems in place when it comes to regulations, prerequisite programs, and food safety management systems, one needs to identify the responsibilities that exist within the food supply chain. Now, you would note from my pictorial that I have a supply chain from farm to fork, but I also have a picture of a recycling logo, as this has become a massive element in our world today, where we find ways to reuse and recycle items. In the food industry, it is very important that you consider risks associated with this activity. And so when you look at the specific diagram, you will note that from a farm level, I have R&I indicating regulatory authority and industry. In terms of the entire supply chain from farm to fork, we have the regulatory authorities as well as industry, that would be you, the manufacturer, regardless of when the process you are, being responsible in terms of making sure that the products are microbiologically safe. The consumer or customer may come into place in terms of when they purchase the product. And how would they come into place in that regard? It is based on information that we supply. So when I talk about based on information that we supply, every element of our food safety management system, from specifications right down to the label that appears on the final product, is there for a purpose, is there to prevent a microbiological risk. So it is imperative that you consider, in terms of microbiological risk, the information you give to a consumer in terms of the label, how to actually handle the product. That is the only information that they can take into consideration in terms of playing their part in microbiological risk mitigation. It is therefore important that it will be understood by your consumer. The same applies to any recycling information that the consumer needs to be aware of, as well as any risk that you may be concerned about in terms of the recycling of recycling information. An example in this regard is, for example, reusable glass bottles that get returned to beverage institutions. The labeling becomes very important in terms of communication to the consumer when it comes to how to handle a product to prevent microbiological risk, as well as where there may be a requirement for recycling and a responsibility that the consumer may have to take. So if regulatory requirements are not clearly defined, if industry, again, do not participate in the process of regulatory requirements to ensure that they are clearly defined and ensure that they're aligned to what may render our products safe, and if consumers fail to adhere to the requirements as put on labels, these are things that can very easily contribute to microbiological risk. In actual fact, some of these elements are the elements that contributed to the listeriosis outbreak of 2018. Now, one may ask, how did this happen? Were there not any regulations in place? The reality of the matter is there weren't any regulations specific to processing meat. What there was some guidance documents and specifications that could be used by industry. But that in itself was not enough, because that regulation or specification did not necessarily identify certain risks associated with serotypes, which is where it is important as industry. We are continuously conducting research, looking at emerging risks around the world, understanding how they have occurred in different sectors, different parts of the world, and taking that into consideration and taking this information forward to regulatory authorities to ensure that we are proactive in preventing risks in our environment. So as RCL Foods, just to share some information of what we did when the outbreak occurred, what you need to bear in mind that although a recall took place on the 4th of March, the signs of a outbreak started in 2017. And thus, as RCL Foods, when information is shared by DOH, regulatory authorities of a concern, every industry is responsible to go back and check their processes to identify if they may have gaps, to identify where they may add additional processes, to identify what more can be done to ensure that this may not be something that is occurring in their facility. I'd like to categorically state that ST6 was not found at RCL Foods. However, to get to that point of making this statement, a lot of work needed to be done to ensure that we were not sitting with the risk, including whole genome sequencing, a very expensive exercise, but something that industry needs to consider every now and then to ensure that there are no emerging risks that may affect the safety of your product in the future. The process in which you apply these controls and the process in which you manage these controls comes down to your food safety management system. A food safety management system needs to continuously look at improvement, and improvement starts with understanding where your risks are, understanding that your risks are continuously changing. Pathogens mutate. As we currently stand, we have the coronavirus that is actually mutating and forming different strains. So we understand the nature of microorganisms, and thus a proactive approach to preventing microbiological risk is continuously researching and finding information, finding various control measures. It is not a one size fits all. In a processing plant where you have heat treatment steps, these are your bactericidal steps where you can eliminate a hazard, ensuring that you may find other processes that may support your prerequisite programs to eliminate the risk or reduce them to acceptable levels. This all starts with leadership and management, as all of these may require, number one, resources, whether it's monetary, whether it's in the form of people, whether it's in the form of consultants. It requires engagement with stakeholders, which includes regulatory authorities, national and international. It requires that we are continuously staying abreast of what is happening out there, and that we are continuously acting where we're finding any anomalies when it comes to microbiological risk. Thank you, Linda.
Thank you for an overall, I think, basis for the purposes of the rest of our discussions today, and highlighting the importance of putting programs in place to ensure that food is safe. I definitely have a couple of questions for you for later in the session, but I think that your initial presentation has provided us with the reason why we should be considering alternatives and reviewing those alternatives to ensure the safety of our product based on where we are in the food chain. Now we're going to present a potential solution, and that being the use of phages. So before we move on to Professor de Vlichera, I'd like to ask you how familiar you are with this particular technology. So Ryan, take it away.
Well, thank you very much. So as with the previous poll, once more, you can click on any of the answers you see in front of you and then click submit in order to participate. The question we have for you this time is, how familiar are you with phage and phage technology for use as a food safety intervention? Your answer options are very knowledgeable, knowledgeable, somewhat knowledgeable, not very knowledgeable, or almost nothing. We appreciate you taking the time to answer. It looks like most of you have managed to submit an answer very quickly. Thank you very much. Let's take a look at the results. Coming in 30% for both knowledgeable and somewhat, 21% for not very, 12% of you have said almost nothing, and then 7% of you are experts at very knowledgeable. Thank you very much for your participation. Linda, it's back to you.
Thank you, Ryan. So I'm probably in the almost nothing section, which means the next presenter I'm going to listen to very, very closely. And so it gives me great pleasure to introduce Professor Frank de Vlichera. He is currently sitting in Belgium, and he's going to be chatting to us today about food safety solution. He has many, many letters after his name, a very highly qualified professor at Ghent University, where he is focusing on food microbiology and food preservation within the Faculty of Bioscience Engineering. He has been a member of the ICFMH committees on food microbiology and hygiene, and he has over 300 peer-reviewed scientific papers. So it's the perfect person, Professor, to talk to us about exactly how phages provide us a solution. So over to you. Thank you so much.
Thank you, Linda, for this nice introduction. Indeed, my talk will deal with how phages can be used as a possible solution for increasing the food safety. Now, first of all, we have to be aware what bacteriophages are. And in fact, a phage is the Greek word for a bacteria eater. So in fact, they are natural enemies of bacteria. And we have to be aware that about every 2 days, up to 50% of all bacteria present in the world are killed by phages. we can say that there are about 10 times more phages present than bacteria in the world, and they are really naturally present everywhere. Just to give you an example, 1 milliliter of seawater or human gut contains up to about 1 billion phages. Yeah? What is important is that these phages are harmless to everything, to every, let's say, living species, except for the specific bacteria that they are acting against. So they are very targeted, they are very specific, they only kill, in fact, one specific type of organisms, like, for example, Salmonella. So these bacteriophages are, in fact, viruses, and viruses with bacterium as a host. Yeah? What they typically do is that they kill this bacterium. So they are bactericidal, not bacteriostatic. So they don't inhibit the growth of these microorganisms, but they are killing them. Yeah? And in fact, one bacterium, or one phage, sorry, can kill one bacterium. But they do this in a very, very selective and specific way. So it really means that all other type of microorganisms that are present in the environment, let's say, where they are living, are not really attacked. They only attack specific species of bacteria. Nowadays, there are phages commercially available against Salmonella, against Listeria monocytogenes, but also others, in fact, are in development and very close to release, let's say, for example, against EHEC. Now, it's important to realize that bacteriophages have two ways, in fact, to reproduce. They have a lytic cycle and a lysogenic cycle. In the lytic cycle, you see that the phage is attaching to the bacterium cell and introduces his genetic material into the cell. And then it is reproducing into that cell until, in fact, as you see at the end here, the cell is lysing and the phages are released. This is the lytic cycle. The lysogenic cycle is different, namely the genetic material is also introduced in the cell, but the genetic material is getting part of the bacterial genetic material, and the bacterium is multiplying, and in this way, in fact, also the genetic material of the phage is reproduced. Now, it is important to realize that the commercial phages that are available are strictly lytic phages. It means they only use the lytic cycle to reproduce, and they don't use the lysogenic cycle. So that is important to realize. Now, I have been looking to the number of publications regarding food and bacteriophages, and you have to say that it is in fact a quite well-established scientific area that already started from half of the 80s, as you see here, and really started to increase from the 90s, early 90s. And you see that it is even, it is not a linear curve, but that in the years 2000, 2010, we really get an increase in the number of publications. So it really can be said that this is an area that has been investigated already for more than 30 years nowadays. Now, focus in this publication is on Salmonella and E. coli, and also on Listeria, and partly also on Campylobacter. So as you see, it is mainly, the big part of publications are on these four of pathogens. Now, what foods are then related in these publications? Well, I also had a look, and you can see that about 44% was dealing with meat and poultry, 35% with dairy products, about 14% with fish, and about 6.5% with vegetables and fruits. Now, it's important to realize that there is some bias on the dairy products, because it is, of course, known that phages play an important role in fact, damaging fermented products. And so their phages are negative because they are disturbing the fermentation process. And most of the publications are dealing with that in the dairy area. So basically, you can say that the top three of products that we are dealing with are meat and poultry, fish and vegetables and fruit. Now, a bacteriophage has in fact a very simple structure. It has a kind of head, the capsid, which is a protein, which is encapsulating the nucleic acid, the genetic material of the phage. And then there is a kind of body and a tail with some spikes. And these spikes are the organelles, let's say, that are recognizing the host and are taking care of the attachment of the phage to the bacteria. Now, this simple structure also makes that we have to be aware that these bacteriophages are not stable in that way, that when they come in contact with foods, that they're going to interact with certain food components, and this can be quite diverse. This can be other proteins or charged molecules, but you will have, in fact, an instability. The bacteriophages will get killed or inactive, in fact, because of interaction with food components. Next to that, also, you have to be aware that certain food processes, like heating, for example, are also killing the phages and also making them unuseful. So this means, in fact, if you look to this instability of the bacteriophages, you should be aware that they are rather unstable when in contact with food. Yeah? And so these phages are neutralized by different interactions with food components, and therefore, in fact, their action is rather temporary and is clearly decreasing with time. This also means that the moment of application of these phages is therefore very determining the efficiency of the process. So we have to be aware at the moment these phages come in contact with the food, their activity will go down. So the moment of application will be very important. Now, how do we apply these bacteriophages then? What is important is that you really have to think about the fact that these phages should meet their pathogen, their host. And they should meet this on time, as these phages are unstable in food. Yeah? So there are, in fact, two bottlenecks, namely the temporary activity of the phages due to instability of the phages in contact with the food matrix, but also the fact that this phage should meet the cell, the pathogen. So, in fact, this phage solution also you are using should reach the pathogen that is present on or in the food. And this is, of course, also from a technological point of view, important to realize. Now, this is illustrated in the following publication where they have applied phages against Listeria on cut melon. Yeah? And what they did, in fact, is that they applied phages 1 hour before Listeria was added to the melons, a half an hour at the same moment as you see here, then half hour later than the contamination, 1 hour, 2 hours, and 4 hours later. And you can clearly see on the right column, after 7 days, so after 7 days of storage, that in fact the reduction is the highest when the contamination happens at the same time than the application of the phage, or when the phage solution has applied before the contamination, and when you apply it after the contamination, that the reduction will be less. So again, here it's clear you really need to pick out the right moment of applying the phage solution. So important to realize is that indeed we get a killing of the bacterium, the target bacterium, not inhibition, and the practice learns that this inactivation is about 1 to 3 log, which means a 90% reduction to a 99.9% reduction of the pathogen. So you can consider this as a kind of light pasteurization, in fact. You are realizing inactivation for one specific bacterium, being either Salmonella, Listeria, E. coli, Campylobacter. These are the 4 pathogens that are most investigated at the moment. Now, I will just show you some results of publications in different type of food products and different type of pathogens. What you see here on the left is the use of bacteriophages against stec, so pathogenic E. coli, on beef trimmings to produce ground beef. And on the left side, you see the control. On the right side, you see the amount of stec after treatment. And you see indeed a reduction, clearly, of about 1 log, I would say. On the right side, you see a figure of bacteriophages that has been applied on salmon fillets against Salmonella. And they have investigated this for different concentrations of the phages, because of course the concentration will of course also matter. And you see that concentration of 10 to the 4th, 10 to the 5th, 10 to the 6th, 10 to the 7th, and 10 to the 8th are applied. And at the highest concentration, you can see indeed that at these 4 degrees Celsius, you reach about 1.5 to 2 log reduction of Salmonella. Another example of another product is fresh cut spinach, where bacteriophages against Listeria monocytogenes are used. And you see the black full lines are the controls and the dotted lines are the treated ones. And you see indeed during time a difference of about, in that case, 3 log. Yeah? So here a rather high reduction was achieved. And on the right side, you see results from bacteriophages used against Listeria also, but on sliced cooked ham. This ham was stored at 10 degrees Celsius. And you also see here a reduction of, let's say, 1.5 log reduction during time. So it's clear you can reduce the presence of pathogens on different type of food products with these bacteriophages. And to conclude, I would say that you should realize that bacteriophages are natural compounds. They are ubiquitously present in nature, and they are very specific. So they only kill the target organism, and they have no effect on other microorganisms or on the background flora. So I'm really convinced that indeed bacteriophages are useful to improve the microbial food safety. It is in fact a well-studied area already. I've been telling that we are studying this already for more than 30 years, in fact, for several food products, for several food pathogens. And indeed, the use of these bacteriophages can contribute significantly in increasing the microbiological food safety. However, what is important to realize is that the way and the moment of applying these bacteriophages is crucial in its efficacy. Yeah? So it should be applied either before or short after the possible contamination. That is important. You have to guarantee that your surface of your food is entirely treated or has been treated entirely. And you should realize that the reduction you get will normally be somewhere between 1 to 3 log when you apply it in a correct way. And that was my last slide, Linda. Thank you for your attention.
Thank you very much, Professor. Yeah, I've got some questions for you for a little later. Can't wait to have a conversation with you about some of the points that you've highlighted. But it does definitely look very exciting as far as a potential solution. It takes me back to second year microbiology, where one of our projects was to build a phage model. I do remember very clearly the structure as you showed it to us on that slide. So thank you so much. We're now going to move on to our next presentation, where we're going to look at the results of some very exciting research that have been done in South Africa, where we're applying phages in the processing of chicken carcasses. But before we get to that, our target pathogen in the case of this research is Salmonella. And so let's understand our knowledge base when it comes to Salmonella as a pathogen in the food industry. So Ryan, why don't you ask us our important question so we can see the level of knowledge in relation to this particular pathogen and the impact that it has on the food industry.
Yes, excellent. It is appearing on everyone's screen as with the previous ones. The question is, how familiar are you regarding the symptoms of Salmonella contamination, the statistics of death, and the cost to the industry? Your answers, as the previous one, are very knowledgeable, somewhat knowledgeable, not very knowledgeable, or almost nothing. Please take a moment to consider your vote. You're all very good at this. Thank you very much for your participation. Very fast. Let's look at our results here. 46% for knowledgeable, 23 at somewhat, 14 tied at both very but then not very, and then 7% coming in for almost nothing. So Linda, it's back to you.
Thank you very much, Ryan. So it's great to see that there are a high percentage of the audience that fully appreciate the seriousness of salmonella contamination within the food industry. And we're going to look now at the chicken processing sector, where it is very much one of the foremost pathogens that are considered. And it gives me great pleasure to hand over to Professor Phageguard, who is a professor of food microbiology in the Department of Food Science at Stellenbosch University here in South Africa. He's the Vice Dean responsible for teaching and learning within the Faculty of Agri-Sciences. The important thing about Professor Phageguard is that he is a South African representative on the ICMFH committee, and he also is the director for the Center of Food Safety, which is a specialist research unit that was founded shortly after the listeriosis outbreak in South Africa. Professor, it's always fun to listen to your presentation, and I can't wait to hear what you and Kirsty have uncovered with regards to this particular research project. Over to you.
Yes, good morning, everyone, and good afternoon for some people, and also a very early morning for Ryan that's actually sitting there in the States. Yes, I think it is quite exciting times now that we're going to work on phages and what they can do with the phages, and it's important for us to share the information that we do have on the phages. So let's move on to the next slide. Linda, yes, at the Center for Food Safety, we are trying to be proactive in what we're going to do in all aspects of food safety. And as Najib and Frank said in the beginning, it's very important to really know what's happening in the industry, as well as knowing your organism quite well. Next slide, please. So Frank also alluded to the number of exciting papers that we've seen coming through the last 5 years, specifically on phages, and it becomes very important to see that what phages are doing in the industry. The early articles were actually a lot of information on how to basically use the phages in a lab setting and before you take it into industrial setting. And a lot of work has been done on Listeria monocytogenes, for example, in coleslaw, Listeria monocytogenes in refrigerated vacuum packed cooked meat products. But a lot of review articles are now coming out of the use of bacteriophages in the poultry industry. Next slide, please. So what we see is that there's actually a lot of information regarding phages and a lot of information regarding the Salmonella problem. Now, through the years, in the early 1990s, we had an incident where PCR was the big thing in food microbiology. Then later on, we saw basically molecular detection systems for pathogens in food samples. And then, as Najib said, whole genome sequencing is becoming popular in understanding the organisms. But what's happening now is that we actually are looking into ways of solving problems. And this, where the phage solution is actually one of the issues that we can use in problem solving. So what is Salmonella? We all know that Salmonella contributes a lot to economic losses in the industry. There's a lot of cases annually regarding this organism, and we see that through the use of antibiotics and growth promoters in animal farming, the Salmonella is becoming resistant to almost all antimicrobials that's out there. And legislation is starting to control what we can do in the industry by, for example, in Europe, we cannot use chlorine, for example, but in the States, you can use that. So as Frank actually said, bacteriophages is actually the natural predator of the bacteria. They're harmful for human consumption, and it's also one of the organisms that's been approved by the FDA. Next slide, please. So what have we done basically in our study? So we first did a lot of research to find out what's been done before we started this research project. So we saw that phages was actually used on chicken breast and skins in a lab setting where they've received a lot of good results. We've also seen that there's a lot of commercial phages out there that's been used in different scenarios with chemicals and without chemicals. And we've also seen that we can use phages together with chemicals as well as an alternative to the chemicals that's been used in the industry. But there was no real study that was done in an abattoir, for example. So the phages were applied within the lab setting, and by using that, they're using micropipettes, so the concentrations differ quietly from what you can do in an abattoir. So we've decided then to basically use a commercial phage cocktail in line in an abattoir on chicken carcasses to upscale the whole scenario of phages to a processing environment. We then also make sure that we observe the effect of the phages that's been applied after the spin chilling step due to the chlorine dioxide that's actually in the water that's there. We then decided to use a much bigger sample size to show if phage is going to be incorporated into a large-scale hurdle concept. And then before we could do that, we had to basically design an appropriate spray system for industrial application to ensure that, as Frank said, that the organism can actually reach the phages, or the phages can reach the organism. So the phages are approved by a lot of different countries. And in South Africa, before we could have started with a trial like that, we had to get approval from the Department of Agriculture, Land Reform and Rural Development in South Africa to ensure that we stay within the regulations that is actually there. So how do we control pathogenic organisms? Well, it's important to note that whenever you want to control anything like a Salmonella, a Listeria, Campylobacter, E. coli, there's three ways of doing that. The first one is to prevent contamination, and that is the most obvious one that we want to do. We want to keep them out of the specific food products or a specific food factory environment. The next one is actually to destroy foodborne diseases by killing them by using different types of chemicals, processing, heat, for example. And then we have the last one, that we can prevent the multiplication of the foodborne diseases. And that's a way that we can control the pathogens. So what did we do in the entire process? Firstly, we had to decide what we're going to do it, how we're going to do it, and how we're going to execute that specific experiment. So we looked at, firstly, the first objective was actually to show there's a reduction of artificially contaminated chicken in the lab by taking the Salmonella that's been isolated from a factory environment to see if the phages can really kill them off. Then we basically had to design a spray system to ensure that there will be suitable application and the phages will reach the bacteria that will be on the outside of the chicken carcasses. And then we decided to take the spray system into a factory environment where we then started to spray the chicken carcasses after the spin chiller at a percentage of 1% over 4 weeks. So what we've obtained from the first set of experiments, we saw that the chicken contaminated with Salmonella enteritidis and treated with a 1% phage showed a clear 2 to 3 log reduction, exactly the same that was actually stipulated in the articles even that was discussed by Frank. The next one is that we had to basically design a spray system because we didn't want to damage the phages as we're putting it through a pressure system that it can be sprayed onto the carcass. So we also do experience that if your pump pressures is a little bit too high, you will have a reduction in your phages, and we had to make sure that the phages will not be killed through that system. We need to make sure that there will be sufficient coverage of the carcass, and it must be part of the process that it can be cleaned quite easily. And we decided that the point of application will be at the spin chiller. So we also know that the chlorine is going to be evaporated in the cold water, and we had to take that into consideration. It's also important that the final stage was the spray of the phages to the carcasses over a 4-week duration. And we worked out that the 10 microliter per square centimeter spray volume will be exactly what we're going to use. And we decided to take neck skin and swab samples to ensure that we'll can detect the organism. And as Najib actually said, we used an ISO method, that's a accredited method for the detection of Salmonella in specific poultry products. And it's important to note that in the States, people are using whole carcasses wash as the way to detect for the Salmonella, the present or absent. And in the EU, they're using 25-gram neck skin samples. So again, going back to the literature, we established that there was no significant difference between the two methods, and you can use either one of them. And then we decided to actually use that specific 25-gram neck skin sample. So the summary of the ISO method is actually that we pre-enrich it in a buffered peptone water solution, and then we basically put it into a Rappaport faciliaris broth at a specific temperature. After that, we put it into XLD agar, and you all will remember that if you see black colonies on the XLD agar, that is the Salmonella that can produce H2S, and that's quite unique for the Salmonella. And then we typically have to do a confirmation from Newton agar, then either to PCR or any other methods that's available. So what did we do? So firstly, we basically spray about 150,000 birds per day over a period of 4 weeks. We sampled the carcasses at the spin chiller for positive or negative. So we're looking at presence or absence in that specific regard. And then we took the spray machine to spray the chickens after the spin chiller, and then we sampled again after the phage application. So I don't want to go into the detail of the exact measurements of what we've been doing and the numbers that we achieved from week 1 to 2, but if we edited the data and we average everything out, we can basically see that about 70% of the incoming birds or the carcasses were positive for the Salmonella. But I'll allude to that a little bit later. And then after the application of the phages, we ended up of 28% positive for the neck skin samples. And if we do a swab sample, it was actually that 1.6% of the swab samples were positive for Salmonella. If you go, can you just go back to the previous slide, please? Thank you. So if you look at the industry norm, that's usually at 35%, you will see that 14% will be positive after the phage application and less than 1% of the swabs. Thanks, Linda, for the next slide. So it's important to note that chlorine have a huge effect on other organisms, but if you have, for example, a scalding tank where the temperature is quite high in the scalding tank, that leads to resistance to Salmonella due to the production of heat shock proteins, but I'll come back to that a little bit later. So if you look at the Salmonella that's been isolated before and after the application of phages, we could clearly see that all of the Salmonella was basically multi-drug resistance, and they were also then able to survive the levels of chlorine that was applied at that specific stage. But if you look at the phages versus chemicals, it is quite interesting to note that if you have a control in a specific environment and you even just use normal water for 30 minutes, you'll have a reduction in the Salmonella count that's there. But if you look, for example, all the other chemicals and compare that with lactic acid, for example, the bacteriophages at a high concentration for 1 to 30 minutes is still the best that can be used in an environment where you have poultry products or white meat products. The next slide, please, Linda. So it is important to note that from previous studies that we've done in South Africa, that Salmonella is building up resistance towards hypochlorous acids or chlorine due to the fact that the Salmonella is forming heat shock proteins in the scalding tank. And if you then use chlorine or chlorine dioxide, the organisms will be resistant to that, and therefore it will go through the process. So let's look at the phage considerations. Frank actually alluded to that there's a few things that you need to remember before you can actually apply the phage. There needs to be sufficient phages required to infect the bacteria. So they must meet each other in the process. If they cannot meet, they will not have any effect on the Salmonella count that's there. So you have to avoid lysogenic phages, and therefore lytic phages is the one that you have to do. And the commercial phage that we've been using, the Phageguard S, and that's an analytic one. So that was also tick all the boxes before we can do that. We also need to make sure that we know what is the correct concentration that can be used in this specific regard, and to make sure that there's not a lot of runoff of water that you add through the spray process, and that was also worked out specifically. And we know that we need to have sufficient contact time between 1 minute and 30 minutes. That's there. We know chlorine is going to have effect on the phages, and before we could have applied the phages, we made sure that the concentration of the chlorine was such level that it will not have any effect on the phages that we applied in that specific regard. So let's look at the conclusion. Firstly, Salmonella is becoming resistant to antimicrobials and sanitizers. We know that, we've seen that not only in the poultry industry, but in other industries as well. We also know that chlorine is a necessity to reduce the number of other organisms in the poultry processing plant, but it's not that effective against Salmonella due to the resistance that can be built up through the process. We also seen that phage can reduce Salmonella on chicken quite effectively in the lab setting, and the combination between chlorine and then the subsequent phage application was quite effective in the trials that we did last year. So we also have to remember that specific pressure pumps will have an effect on the phages, but if you can control that quite effectively, you will see that there will be no detrimental effect on the phages, on the pressures that you've been using and the concentration that you've been using in your spray systems. So at the end, the results have shown that the phage can be used in the South African abattoir and have a high efficiency in the reduction of the Salmonella that's basically in line. So we've seen from the commercial one that we've been using, it was a smart choice because it's actually been used before. It's been even tested against some of the USDA's top 10 most virulent Salmonella that's there. We've seen it was actually quite a green choice that we've been taking because it was specific for the killing of pathogens like Salmonella that's there, and it does not require any labeling on your product, and for worker safety is one of the best that you can use, and it was also quite easy to apply it through the spray system in line, but if you have to go into more detail and even if you want to have a better reduction of the Salmonella, you can also spray it into your final product or at other stages in the processing line. So yes, I need to acknowledge my students that were working quite hard and doing all the sampling and everything that was done accordingly. I need to acknowledge Stellenbosch University for allowing us to do this specific experiment in the labs that we do have. Again, the Department of Agriculture, Land Reform and Rural Development, Phageguard and Micros for supplying the phages that we can actually do the big experiment, spraying systems for helping us and designing a spray system, and of course the abattoir that we basically could have used in this specific regard, the management for the foresight and using phages as one of the methods to reduce Salmonella, and all the staff and basically the quality control people in that specific environment. So with that, I'm open to a lot of questions.
Thank you very much, Prof. Very, very interesting results, and I'm sure we would like to explore them a little further. We're going to now go to our next poll, because given the information that we've now learned about phages and the amount of research that has been done, plus the practical application of this product within South Africa, we'd like to understand whether or not you would consider phages as a kind of insurance product to produce safer food. So why don't you give us your opinion based on Ryan's last poll. Thanks, Ryan.
Yes, thank you. Yes, for the last poll that we have for everyone today, as Linda has said, the question is, would you consider phages as an insurance product to produce safer food for the end users while reducing the risk of claims and recalls? Your answer options are yes, no, maybe, or need more information. We'll give you a few more seconds to submit your answer. Thank you everyone for your time and for participating today. Let's take a look at where the results have landed. It looks like we have 63% for yes, 19% for need more info, 10% at maybe, and 8 at no. Linda, that's back to you.
Thank you, Ryan. Our last presenter today is Mr. Isak Breitenbach, and he's the General Manager of the Broiler Organization for the Southern African Poultry Association. Unfortunately, Isak was not able to be with us live today due to other commitments, but he has recorded a message from SAPA with regards to their response to this research. So Ryan, would you mind sharing that video clip with us?
Linda, thank you very much. I would like to give you an introduction into the South African poultry industry and also illustrate where I see opportunity for phages to play an important role in reducing or improving the safety of poultry products in South Africa. The South African poultry industry invests heavily in technology, and that has made us globally competitive. Housing technology we adopt from Australia. We have huge houses in South Africa, 50,000 birds per house. That reduces the unit cost of production tremendously. Secondly, in terms of livestock production techniques, we adopt our livestock production techniques predominantly from Europe and America. In terms of abattoir technology, I don't think that if you stand in an abattoir in South Africa, that you will realize that you're not in Europe. We are using European abattoir technology and is totally up to date with the latest technology being used in Europe. Product development is very important for us. We have a number of markets that we serve. We serve the low-income market. We serve the local high-income market. We serve export markets, and product development is paramount in the South African poultry industry. The South African poultry industry is the sixth most competitive producer of poultry meat globally. Sometimes we are actually fifth, depending on the other countries. We have outperformed the European Union for the last 13 consecutive years in terms of low-cost poultry production. And we could only achieve that by investing in technology like what we talk about today. The world around us is changing. The whole world is moving away from antimicrobials and antibiotics. We move to alternative remedies, specifically in the European Union, but also in South Africa. We are not far behind Europe. With the drive to export more poultry products, this process is intensifying in South Africa. Focus on products like probiotic and phages that is safer for human consumption is the future that we would like to invest in. Phages fall into a new category of product that make food safer. We can see that this product application is an additional quiver in the armor of the producer in fighting Salmonella and Listeriosis. Salmonella and Listeriosis are gut bacteria that's prevalent all over the world. And we certainly all suffer from incidences of these diseases. And this product will greatly assist us in making food safer in South Africa. Phages is not a new technology. It is a technology that is globally available. Production units exist in America, in China, as well as obviously in Europe, that the products we talk about today. But this product has never been operationalized in a high density slaughter environment. The Bureau for Food Safety at Stellenbosch University has done extensive trials to prove that this product is actually working in the South African environment. What was more important, or is more important, is that we need to be able to prove that this product will work at all times. And therefore, the protocols developed is essential in making this product available for poultry producers in South Africa. The potential that we see for product application are various. We talk here about organic and free-range production, where antibiotic use is not allowed. And certainly the product will fit and be an important instrument in the hands of the organic or free-range producer in the future. Fresh poultry meat tend to have higher levels of bacteria simply because of the fact that the product is not frozen and the carcass temperature is a lot higher. And in this particular aspect, there's a huge opportunity for phages to make this product safer. Phages are not only relevant in fresh chicken production, but also in frozen chicken production. Frozen chicken production locally, but also frozen chicken production for export purposes. A country or a company cannot afford to do damage to its reputation in exporting a product that could contain either a salmonella or listeriosis. If we then summarize and talk about this product almost acts as an insurance product. Not only an insurance product for the producing company, but also an insurance product for clients. If a client feels uncomfortable with product supply into its operations, like a retailer or a fast food outlet, they can use this product and safely ensure that the product does not contain salmonella or listeriosis, depending on the product that is being used at that point in time. You can certainly see that if you talk about the South African environment, phages can greatly enhance the food safety aspects of meat in a country like South Africa, a warm country where bacteria growth is higher than in the colder countries in the world. Thank you, Chairman.
Thank you, Ryan. And thank you, Isak, for those words, just with regards to how this product can assist the poultry industry within South Africa based on the research. It's now an opportunity for you to place your questions. We have a couple of questions which I'm going to start with in the meantime. We are unfortunately running a little over time with the technical difficulties that we've had, but I would still like to pose a few questions to our experts while we have them on the line. So thank you, Frank, and could everyone follow Frank's lead by putting themselves back on camera so that we can ask the questions to the panel. Professor Najib, if you can, otherwise we know you're in the background. Perfect. Right, the first question that I have today is quite an interesting one. Prof. Gous, I just want to confirm with you, in this case we have a veterinarian who is asking a question with regards to chicken with skin or without skin. So if I understand you correctly, in terms of the work that you have done, we were still referring to carcasses that had not been processed. So we're actually referring to the bird after defeathering, but still with its skin on. Am I correct?
That's 100% correct, yes.
Okay, right. And I think in terms of the further processing, the application of the phage would be after the product has been processed, and you would then be applying the phage solution onto the final product prior to packaging. Would that be how you would consider using it in further processing it?
Yes, I think that is one of the things that I alluded to in my talk, is that where we actually applied the phage, we thought that was the first point that we need to control the salmonella in the abattoir that will allow us to do it by using the phage. But I think if you're going to move it downstream and even just before packaging, you will have a huge effect on the incidences of salmonella.
Professor De Flikkeren, would you like to comment on that? Because obviously the review that you've done of all the applications would consider where the phage is applied, as you also pointed out, is a very important point.
Yeah, what is important is that you apply the phages solution very close to the contamination. So indeed, also during further processing, there is always some contamination happen. So therefore, it is also beneficial, I think, to treat just just before packaging, because then all contamination has happened, and at that moment it could be very beneficial to apply it. Indeed, yeah.
Absolutely. Professor Gous, this is probably one of the questions that you've been anticipating, and that is, do the phages control the biofilms? I mean, I know biofilms is probably your second most favorite word. So how do we consider, the application of phages when it can, or do phages have any efficacy when it comes to biofilm production?
Yes, it's been known through literature that phages do have a influence on biofilms, specifically within a biofilm, because it's quite interesting that the phages can infect the bacteria. The bacteria will basically produce more phages, and those phages will actually infect the other ones that's also in that specific biofilm. Yes, it will do so, but as you know, within a biofilm, there's also some other bacteria associated. We don't have biofilms that usually just consist of only Listeria or only Salmonella, and therefore some other techniques you have to use as well.
If I understand correctly, you were specifically targeting Salmonella, and so as Professor De Ficker said, the phages are specific. So if I had a double whammy problem and I'm sitting with Listeria as well as Salmonella within my plant, would I be correct, Professor De Flicker, in saying that I would need to use more than one product in this situation?
Indeed, then you should use, let's say, a mixture of two phages or phage solutions, one against Listeria, one against Salmonella. But of course, you have to be aware, Listeria contamination is very often happening on a different moment than Salmonella contamination. So it could be beneficial to apply it in different moments also. So it's not necessarily the best to mix, let's say, both phage solutions, but I can imagine that it would be better to apply it specifically on a specific moment for Listeria and for Salmonella. Yeah,
Perfect.
but indeed you will have to use two solutions then. Yeah.
Now,
maybe if I can just add there that the one will not interfere with the other. So even if there's two different phages in a specific same environment applied differently, the two will not interfere with each other.
I'm now going to hand back over to Ryan. Unfortunately, we've run out of time. I would have loved to have asked a whole lot more questions, and I'm sure you would have too. But Ryan, would you like to wrap it up for us? Thank you so much.
Well, thank you very much for that insightful presentation and for those wonderful answers. However, yes, we have reached the end of our time here today. Though if we couldn't attend to your questions, the team at Phageguard may follow up with you. Or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've sent you a link in the chat box where you'll be able to view a recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join us once more in thanking our speakers for their time here today. We hope that all of you found the webinar informative. Have a great day, everyone, and thank you for coming.